"35 years ago, when I joined the biology department here at Princeton, there was a profound change taking place in the subject, even though many of the professors continued to make classical investigations into higher animals and plants. The building is still packed with dinosaur skeletons, bones, collections of stuffed animals, and many other treasures accumulated by enthusiastic collectors doing a long and distinguished history. These pictures, which illustrate the evolution of the amphibians and the reptiles, are particular favorites of mine, and they were painted nearly 100 years ago by an eccentric Englishman. I always have a very special feeling about this room. This is just down the hall from my lab, and it belonged to Mr. Charles Rogers, who was the bird curator. Because this is the way I began. When I was about 10 years old, I became fascinated by birds. And what I wanted to do, more than anything else, was just study birds. And this is the sort of heaven that I had in mind. And I also dreamed of having drawers of birds, the way you can see here in these wonderful cabinets that have all these bird skins. And it seemed to me that just collecting birds, identifying them, was just absolute bliss. But my father was very clever, because while he thought what I was doing was fine, he also thought that I should broaden my view a bit, so he gave me for Christmas a book in biology. And that did the trick, because I started reading it and was just tremendously interested in it, and began to start thinking about bigger problems than just birds, and such problems as evolution, for instance. And then ultimately the one that really gripped me was the problem of development. Growth, especially of embryos like this human fetus, is the subject matter of developmental biology. Look at all the complicated detail. How did the hands and fingers, the face and the brain grow and form from just one fertilized egg? I wanted to find out, but I wanted to do experiments as well to test my ideas. It was only when I went to the university that I could see perhaps the best way of doing this was to study some simple organism. And it was then that I chose slime molds, and I've been working on them ever since. It's a remarkable thing that if you scrape up a bit of soil anywhere in the world, you can find lots of tiny creatures in it. Upstairs in the lab, I mixed the soil with water and then spread it on the surface of an agar plate. Agar jelly is full of nutrients, so after a couple of days in the incubator, the beasts in the soil have grown enough to be quite clearly visible. Now this plate has the soil on it spread in a suspension, and I'm going to place it under the microscope here. This wiggling object is a nematode, a kind of lowly worm that is a voracious predator in the soil and eats all sorts of things. This rather horrible-looking creature is a mite, which is closely related to spiders. It is a very common organism and found not only in the soil but also in cheeses. When you see a small organism zip across the screen, that is a protozoan, which is a single-celled organism, very common in the soil and also in ponds. These filaments are fungi, and they're extremely common. One finds them everywhere. For instance, on stale bread. If I speed up time by photographing this scene through a movie camera, by taking one picture every 30 seconds, and then look at the movie, the whole scene looks remarkably different. Seen this way, easily the most striking organisms are a sort of tiny, glistening forest. These are the slime molds. Slime molds are really quite remarkable organisms, unlike almost any other kind of organism. They consist of tiny amoebae, and these amoebae are very small cells. They live in the soil and they feed on bacteria. And after they're through eating and have completely wiped out an area of bacteria, then they don't just sit there and wait for more, as you'd expect, but instead they do this extraordinary thing of streaming together into collections. Now, these collection points consist of masses of cells. They may be 100,000 amoebae or so. These cells now take on a shape. It looks like a slug, and in fact it exudes slime out of its back end, so you can see where it's been. And it now wanders around for sometimes quite long periods of time, up to a matter of days it can wander. They do look quite slimy, and in fact it's probably because of that that the early German workers thought of them as being slimy mushrooms of some sort, so they called them slime molds. Indeed, there is a really interesting question which one could ask, and that is, this seems a very primitive multicellular organism. Is it, therefore, our ancestor? Is this the way we became multicellular and evolved from amoebae? And the answer probably isn't quite yes or no. The answer is that in primitive organisms, there seem to have been many different attempts to become multicellular, and this is just a very curious and interesting one. You can put it like this. The first life on Earth was all single cells. It was only much later that these cells aggregated together to form multicellular organisms. One such primordial event led to the plants, another to the animals, including, of course, us. The slime molds, however, seem to have got stuck, endlessly repeating very ancient history. One of the things that many, many people have asked me is why on earth have I decided to really spend all my laboratory life working on slime molds, and what possible interest could this be? And there are, of course, many answers to this, but let me just say that they do certain fundamental things, and they're very easy to study. And so one can actually learn about those fundamental things more effectively with them than often one can with more complex, higher animals or plants. Let me give you two examples. For instance, there's tremendous interest in our own bodies, the fact that our white blood cells are forever eating any bacterial infection that comes in. Well, slime molds spend a good part of their life eating bacteria, engulfing bacteria, so that this becomes a very effective way of studying this really quite fundamental problem. These are in fact phagocytes, or white blood cells. They look exactly like slime molds. The parallel between the two is really very remarkable. Some years ago, an old friend thought that perhaps this might be of some great medicinal use, and he tried to see if he could get slime molds to grow on wounds to help clear up bacteria. But unfortunately, slime molds don't grow at body temperatures, so this great plan never worked. One of the first things I became interested in slime molds was the business of aggregation, of the coming together of the cells, because this seemed like, A, such a nice thing to study, and also it is a rather fundamental problem in biology. And it was particularly intriguing because at the time there were many people who felt that it was too difficult to study on any organism. And what I found out very early was that this was due to a chemical which is attracting the amoebae. Now, it's very much the way a bad smell will attract flies, so that the amoebae are like individual flies, and somehow they can sense that there's more of this chemical in one place than another, and so they stream towards it, and instead of being bad, they love it. The next really exciting step was the discovery of what this chemical was. And this actually took place in this laboratory with the help of some colleagues from abroad. And what we did was we found, quite by luck, that this substance that was responsible for this was a substance known as cyclic AMP. Now, this is a very fashionable substance because it turns out that we're loaded with cyclic AMP. It's involved in all sorts of hormone reactions in human beings. It's involved in the action of the brain cells and so forth. It's an extremely important chemical, and here is this same chemical being responsible for the aggregation of the amoebae. And we could show this by, once we found out what the chemical was, we could show this in a very simple fashion. What we do to do this is really an extremely simple experiment. What we do is to take an agar block on this petri dish. Now I have some agar jelly. And now take a simple, cut off a little block of this agar jelly, and I lift it up into the air. And now this agar jelly I saturate with cyclic AMP at just the right concentration. And now in this plate I have some aggregating, or cells that are about to aggregate. Now here's the attractant. And I put this in here right at a spot where they're going to be most interested, because there now is the attractant. And what I do is you can put this now under a microscope. This is not a time-lapse attachment, so I can take movies of the process. And as I look through and get things in focus, we can now, through watching this over the movies for a long period of time, actually see the cells just stream into this agar block in a very dramatic way. In this shot, on the left-hand side, is the edge of an agar block which contains just the right amount of aggregation attractant, the chemical attractant. Now, what you see is a group of rather young cells, and clearly by the band that goes across, you can see that they're affected by this attractant. But it's even more dramatic if you take cells which are just at the right stage, and then this attractant is so powerful that all the cells start streaming in towards the agar block, and it works so well that ultimately you begin to see slugs forming normally. There is the interesting question now as to exactly how the chemical does this. And without going through any of the complications, it really amounts to this. You must think of an amoeba of having a nose at both ends. And if the nose at the front end gets more molecules hitting it of this attractant than it does at the posterior end, then it knows that it should go in that direction. So that what it is constantly doing is it seems to be taking readings at its anterior and posterior end and deciding where the chemical is more concentrated. And it's very much the same as we would do if we want to locate a smell. We would take a whiff here and a whiff there and then decide that this one was stronger than that one, and that would give us a guide as to where the bad smell or good smell comes from. We unfortunately do not have noses at both ends, and so we can't be quite as clever at it as amoebae can. The aggregation cells don't move in smoothly. It is as though you dropped a stone in a puddle and there was a ring going outwards. And this outward ring actually is a ring of fast inward movement of the amoebae. Now, apparently what happens is that this cyclic AMP, this chemoattractant, actually stimulates the cells to produce their own. So you have sort of a relay of passing of this response and stimulus, and it really looks quite remarkable, as you can see. Years ago, when I took an ancient film of this, and it showed these waves beautifully. At that time, Professor Einstein was living here in Princeton, and a mutual friend told him about the film. And so he asked me if I would be willing to show the film to Professor Einstein. So I, of course, said yes and took it around to his house. And we went into his bedroom and took a map of the USA and turned it backwards for a screen and pulled the blinds, and I showed him the film, and he could not possibly have been more delightful. And he asked me all sorts of deep questions about what these animals were really doing and what was going on here. The thing that I remember particularly is that that he was very kind to me and very interested, but that there were other people there who kept feeling that somehow he wasn't understanding what I was saying, and they would try to explain to him what I just said, and he would suddenly turn around and sort of be furious with them for interrupting and asking such or making such stupid statements. And the fact that his accent, his appearance, and everything was exactly like my grandfather. So I had this feeling suddenly again of being in my grandfather's office and watching him tear people down for saying stupid things. Slime mold amoebae have an inclination to move. These cells have just aggregated, and they are blindly chasing their tails, running around in circles. Eventually, the circles turn into migrating creatures that crawl away. They produce this extraordinary structure that looks very much like a garden slug. And in fact, in slow motion, it moves like a garden slug. Of course, it's tiny. It's only a millimeter or two long. But it is, on the other hand, not a garden slug. It's just a mass of amoebae. that despite this simplicity, the remarkable thing about it is that it behaves like a multicellular organism. It is, in fact, so extraordinarily sensitive to light and heat that the amount of light needed to attract it, the dial of a luminous wristwatch would be quite adequate. And the heat is perhaps even more remarkable. These things only move about 1 millimeter an hour, so no one has had the patience to carry out this experiment. But presumably, if you stood in one place long enough, people give off enough heat so that you would slowly have all these slime mold slugs crawling towards you. This, however, is not perhaps a practical experiment to do. Each slug constantly produces slime as it crawls along, but it leaves that slime behind so that it is crawling always out of its sheath. And so what you're seeing here is an empty casing which is left behind. So this gives you the impression that slugs really are quite remarkable unitary organisms. They really are a multicellular organism, even though they've arisen by the aggregation of these very independent amoebae. As it crawls along, each slug is preparing itself for a remarkable metamorphosis. Sooner or later it stops, and astoundingly transforms itself into something rather like a minute plant by propelling itself off the ground on the end of a stalk about 1 millimeter high. In the tiny world of slime molds, gravity is not an important force, so even humble amoebae can launch themselves up into the air. The secret of this transformation lies in changes that take place inside slugs as they crawl around. I think these slugs are remarkably like embryos, except that they are on the move. So I think of them as motile embryos. In this respect, it's really quite unusual because mammals don't have motile embryos. And it has, therefore, many properties of an embryo. And one of these properties can be demonstrated very easily. You can take a slug and literally mash it up. So you have a rubble of cells. And then you just wait a short time, and those cells come back together again and reform a slug and wander off. And they seem to be completely unaffected by this mistreatment. Amazingly, when we are very young, just a few days after conception in our mother's wombs, we too can recover from being mashed up into a rubble of cells, just like these slime molds. There are other things that they do that are remarkably like embryos. For instance, embryos of mammals can twin. You can have a small embryo, particularly in some species, which form, and then that embryo divides in two so that you have two embryos. Well, the slugs do the same thing. You can see beautifully that very often a slug will be crawling along and suddenly the thing will separate into two slugs of similar size. This is the same thing as twinning, and in fact, one can see beautiful examples of such twinning in mammals, which very closely parallels this kind of situation. Perhaps I can illustrate the connection between embryos and slime molds by showing you some more of the many treasures stored in this biology department. Now here we have an ancient collection of embryos, and the reason for my showing you these is the fact that they represent development. And what I mean by that is that they show that almost all organisms start off as an egg. And then this simple, or at least apparently simple cell, turns into an incredibly complicated structure. Look at this row here now, where here are different stages of the growth of an opossum. And these many offspring, which you see finally in the last stages over here, all came, each one came from a single egg. And the same thing is true in the case of the kangaroo down here, the sharks. Here's a lamb, and here's a fetal pig. This is the fertilized egg of an amphibian. One cell grows into a ball of cells by a series of divisions. This is extraordinarily similar to the development of other animals, including humans. Soon the cells start to migrate around in carefully orchestrated and complicated movements that finally create the correct form, in this case, of the toad that will eventually emerge. During the course of this development, you have, this single cell has produced an incredible structure. It involves all sorts of different cell types. It involves muscle cells, it involves nerve cells, it involves not only that, but the arrangement of these things. I mean, think of myself, for instance. I'm full of muscle and nerve cells, and the extraordinary thing is that the proportions of my arms to the rest of me are roughly the same as are those of any other human being, so that this same proportion goes on from generation to generation. So this is really a very complex process, and one wants to know more about it. Well, my idea has been that perhaps one way we can approach it is to look at a very simple organism. And the simple organism, of course, is the slime molds. Let me now draw some parallels between the two. In the case of the slime molds, when they begin forming a slug after aggregation, and if you do a little trick of staining this slug with a vital dye, then at first the slug appears uniformly stained, but after a little bit of time, the front end is very dark red, and the hind end is quite light. And now we know that those dark cells in the front are in fact the cells that are going to turn into the stalk, while the hind cells end up right on top of the stalk as a mass of spores. It's entirely similar to the development of a more complicated embryo, but instead of hundreds of cell types, you just have two. So we seem to have a perfect sort of micro example of differentiation, and one that should be very much easier to study experimentally. And that's the reason we do it, because we all agree of the fundamental importance and exciting nature of the problem of how an egg or a single cell turns into a complicated multicellular one. When I first started working on slime molds, I thought this would give me a very distorted view of life, because after all, the slime mold is not quite the same as a human being. Then I did, at those early days, work on other organisms as well. But now I have come to the conclusion that actually it was an enormous help to me to look at the world through the eyes of a slime mold, so to speak, because by doing so, I think that it has given me a kind of perspective for bigger problems that has helped me enormously in my writing, and especially my writing of books. A few years ago, a group of Russian scientists came through. So I explained to them how these amoebae are collective and congregated to form this unified mass. That was just absolutely bliss as far as they were concerned. They were very, very excited. However, I ended up by telling them that those cells that got to the front of the slug once it formed are the cells that eventually turn into the stalk, and they die. So that there clearly, in the case of slime molds, a certain disadvantage in being the ones that get out in front, because you end up by being altruistic for all the others. And they did not like that quite as much. One of the things that always seems to me amazing about slime molds is the fact that you do lose a large number of cells in the stalk, in the dead stalk, to form one of these fruiting bodies. You lose 25, 30%, something of that sort. And when you look at it one way, you realize that those cells are actually sacrificing themselves for the spores. So it really is a case of sort of cellular altruism, and they're dead, they're gone, they're not recyclable at all. But if you think about it a bit, you realize that that is not just a peculiarity of the slime molds. That's really a very general principle. In fact, it is a sort of basic strategy that you find among all organisms. For instance, a stalk of wheat grows, and at the top there's the seed, and then in this late summer it turns beautifully yellow, and that's because it's all dead. So again, you have in this plant this long stalk made up entirely of dead cells, and at the top you only have the viable seed. So that it's, in fact, you can think of almost all organisms have this same principle. Consider ourselves, for example. We do it in a much more lavish fashion. Practically all our cells are sacrificed in the sense that the strategy is always to see that some of our cells can go on to the next generation. So in the case of the slime molds, the strategy is to make spores that go on. In the case of the more expensive case of the wheat, it's only the seeds that go on to the next generation. In our own case, it's only the very few reproductive cells which go on to the next generation to produce perhaps 3 or 4 children at the most. So death, from the human point of view, may not be the extremely distressing thing that we think of it if you compare it to what happens in slime molds. Because then you can see that it is really part of a strategy. And this strategy is something in which manages to perpetuate one generation to the next through certain cells which are protected and can manage to produce the subsequent generations. Now, this fruiting body is really sort of like a plant. It has a stalk to it, and that stalk holds a mass of spores at the tip end. But how does it get to the next generation? There they all are, stuck in clumps high up in the air. So what happens next? The spores are covered by a sort of glue, so they stick easily to any passing creatures and can be carried far away. If they are taken to a place where there are bacteria again, then they split open, and out of each spore comes an emaciated single amoeba. And this single amoeba will immediately start feeding on the bacteria and get fat and divide in two. And in this way, you start the whole cycle all over again. You must picture these organisms growing in little cavities in the ground. And it's thought that undoubtedly what happens is that small insects and nematode worms and various things of that sort slither through these holes, and the spores stick to them, and then they carry them elsewhere. And now there's a little bit of evidence that this might be exactly what happens, and that evidence comes from these really intriguing experiments with gas orientation. it's clear that a fruiting body just doesn't rise up in the air without any kind of cues, any kind of directions. It's a very well-oriented movement. Now, it will go towards light, and that affects it to some extent, but much more interesting is the fact that it is producing, as it rises up in the air, a gas, and this gas is apparently a repellent, so that it tries to get as far away as possible from the repellent. And you can show that in a number of very simple experiments. You can, for instance, put two fruiting bodies right close to one another artificially, and then they will, when they go up in the air, they will bend way away from one another. Or you can put one in a little cavity, such as imitating a cavity in the soil, and then you can see that it rises so that it's absolutely in the center of the cavity. It's very good at gauging the sides simply by this repellent it puts out. We had a grand time looking into these repellents because of the fact that we built the world's smallest wind tunnel. And we're able to show that when you, with the help of the aeronautical engineers here, and we could show that when you put these fruiting bodies in the wind tunnel, that they would actually lean into the wind. And the reason, of course, being that the repellent was removed from the upwind side and pushed posteriorly or behind it, and in this way the fruiting body actually moved towards the wind as it rose up into the air. That experiment is more fun than really definitive. A definitive experiment was to use activated charcoal. And we put a little bit of activated charcoal near a rising fruiting body, and now that removes the gas, it absorbs the gas. And when you do that, you see the rising fruiting body actually dive into the activated charcoal because you've removed the repellent from that side of the beast. So that this strongly suggests that indeed in the soil cavities they do manage to make some very nice orientations so that they can get picked up by passing insects and so forth. This is a different species of slime mold, Polysphondylium violaceum, which culminates in branches. Each branch is a perfect illustration of how fruiting bodies grow apart. Sue.
Yes.
Interrupt for a second? I got two burning questions to ask you. One is, have you fed the birds?
Yes, I fed them.
Good.
I watered them.
And the other is actually, you know, we ought to come to a decision about when you have your committee. When are your experiments going to be all set so you can do that?
Well, I found out, I think yesterday, that it doesn't matter when my experiments are set. I have to have a committee meeting because I'm in the last half of the alphabet in March. But
the only really uninteresting projects are the ones who are done by students who really don't seem to have very much imagination. But I think on the whole I've really been quite lucky and had an awful lot of people who've come in and who've done things on their own ideas, that have turned out extremely well. And I think that it's a basic principle which I hold to, and that is that the way to do research work of the sort that I am interested in is to just start doing things.
I'm trying this staining technique of Sumino, and it doesn't seem to me that the bacteria are taking up the dye, so I'm trying to get blue and red.
They look green.
I know, and orange.
Well, there are a lot of wonderful people that have worked in this lab, and a number of interesting stories have come out of this, and one of them has to do with birds. It so happens that my laboratory assistant is a very keen bird bander. And so she was wondering whether or not birds which she'd banded various places in New Jersey might have slime molds in them. And she discovered that they do indeed. And not only that, but she's really done this beautiful bit of work, all on the side, as it were, in which she's shown that these birds carry slime mold spores all over the world, and that they carry them for long periods of time. These birds here, she used to find out how long a bird can hold the slime mold spore in its gut. And they can keep them up to 6 days, and the slime molds are able to live after all that mistreatment. Now, another good example of the sort of thing that's wonderfully pleasant and surprising is a work of a postdoctoral fellow who got interested in bats and whether they might carry the spores of slime molds. And so he had this idea that what he ought to do is he ought to get bat dung from inside, way inside a cave, a place that never saw light and was at constant temperature and humidity. And so what he did was he collected some of this. His idea was sort of interesting. He thought maybe there's such a thing as a blind slime mold, the way there are other blind cave animals. And what he meant by that is a slime mold that would not go towards light. Well, he didn't find that at all, but he found something much more interesting. What he found was, and for reasons which are not important, he decided to try and mix different species that he found in the cave together in the dish to see if they aggregated together or what. And he discovered, much to his amazement, that there was one particular one that no matter what he mixed it with, it was the only survivor. The other slime mold always completely disappeared. So he then became very interested in that. Did they produce a poison, or how did they do this? And what he discovered is that this slime mold ate the other slime molds, so that it is a predatory slime mold, which was completely unknown before. And he has called it, the species name he gave it rather cleverly as caveatum. This caveatum now has been intensively studied. And let me just give you an idea of how dramatic it is. If you take very few cells, so let's say 1 in 10,000 cells, and add it to an ordinary slug of the species we work on most of the time, then that slug will crawl along as though nothing were wrong at all, absolutely happily. And then suddenly it stops in its tracks and begins to look a little peaked. And then after a period, there's this great bloom of beautiful little fruiting bodies that come out of the body, or the carcass, of the slug. Caveatum is not a parasite. It's a predator, like the tigers and the sharks. The difference is that caveatum eats its victims from within and then bursts out of the carcass. You must imagine it living in its dark, constant temperature caves, waiting for the bats to flutter in with their loads of dung, rich in its slime mold victims. One of the things that we couldn't help wondering about is if caveatum is such a voracious predator, why doesn't it take over everything? In fact, we were even concerned that it might attack humans, but I'm happy to report that that turns out not to be the case. But more seriously, why is it not the only slime mold left on the surface of the earth, since it seems to be such an effective predator? And this question is a very, very interesting one. It's one, in fact, which we don't know the answer to. I think it's a perfect illustration of the fact that every time you discover something new, then immediately there are all sorts of these subsequent exciting questions that one wants to dive into. Hi, so is that the experiment that you were telling me about yesterday? Mm-hmm. Of course, if you are a molecular biologist, which is very fashionable right now, the chances are, if you're doing a good experiment, there may be 1, maybe 5 different groups all over the world that are doing the same thing, and it's a sort of a race. And I just can't stand this. I much prefer to be doing things which might turn out totally new and unexpected principles, things that nobody else either has the wisdom or the stupidity to be doing. When I was being inducted into the Army in the Second World War, and you have to go through this dreadful business of being processed, and I finally got to a desk where there was a burly sergeant with a huge volume in front of him, and he said to me in terrifying tones, "What do you do?" And I said, "I'm a biologist." And so he looked in his book, and he said, "There's no such category." And I said, "Well," I said, "I'm still a biologist." And so he said, "Wait," and he got another book, and he finally found biologist in it. And then he looked it up, and he said to me, "Are you skilled or unskilled?" And I explained to him that biologists are not classified that way. And so then he said, "Well, now, how many years have you been doing it?" And I said, "Oh, about 6 years." And he said, "Well," he said, "if you're not skilled after 6 years, you must be no damn good."